All right, great. So I want to thank the organizers for the opportunity to share a little bit. And it's really nice to follow Dr. Meyerhofer and Dr. Hillegren, because I'm going to talk a little bit about some imaging questions and how the metabolism of immune cells is affected. I happen to be a kidney cancer-focused cancer researcher, so that's why the kidney is on here. Just so we don't all forget about kidney cancer. So I actually want to start with my disclosures. They're mostly my husband dabbling in various ways of losing a lot of money. But my biggest disclosure actually is my collaboration with him. And we just came back from our weekend retreat with the combined Rathmell squared laboratories. He's an immunologist who studies T-cell metabolism. And what I came back away from this meeting was that we really should spend a lot of time thinking about non-cancer people. And I learned a lot about that. I don't think we understand really what the basic of the human immune system is, like what is normal. So I'm not going to talk about that today, but I really hope we can talk about that in the extended discussion tomorrow and maybe right after this. So, I think when we think about biomarkers and surrogate biomarkers of the immune response or resistance, we really, at the current state, are mostly just at the tip of the iceberg. Most of our biomarkers really being tissue-based, which of course leaves all of the unsampled pieces of the tumor, as well as the host. It ignores even tumor heterogeneity within an individual tumor. It misses some metastatic sites, and it loses entirely dormancy niches. So I think what we really have to think about sort of this bigger context as we look at experiments and policies that might help us really get on top of resistance. So what are we looking for in an ideal surrogate? Something that's informative of the holistic disease state, or the state of the patient in that particular state and time, that's accessible repeatedly and using a minimally invasive modality that can help define that host immune response and is a reliable estimate of tumor response and can guide treatment choices through successive treatments. We're talking now about second-line therapy, but there will be third and fourth, and ideally more personalized and tailored immunotherapies that knock things out once and for all. So the story I decided to tell, rather than try to survey what's out there in the many surrogate immune markers, is a story that we published about a year and a half ago, looking at imaging the immune response. And this came from a question of me being a cancer biologist and thinking about PET scans, and PET scans reflecting the cancer glucose uptake, cancer cell glucose uptake, and living with this immunologist who said that, in fact, T cells really want the glucose, and it's important for the T cells to get the glucose. And we know that if you do a PET scan in a patient there on the left, that's a patient with lung cancer, and we can track very reliably lung cancer with FDG PET scans. But if you look at a patient who just had a COVID-19 vaccine in their deltoid, their PET scan will also light up, right? So both kinds of cells will have this preferential glucose uptake. We already talked about in a prior talk what a conventional FDG PET scan is. And we also know that when you treat a patient with immunotherapy, that there can be these wildly confusing responses. Here's a patient with metastatic cancer who has a pre-treatment PET scan and then a post-treatment FDG PET scan after they receive immunotherapy with this blush of tumor signal. And is that tumor growth? Is that pseudoprogression? Is that evidence of immune response? And how do we know what that might be? So in our laboratory, we decided to really try to understand, who really is getting to take up the glucose, right? Was it the cancer cells? Was it the T cells? Was it somebody else? And that seems like a very simple question, and it actually was. It was laborious, and it's a very difficult experiment to do, but is conceptually quite simple, which is that we did FDG PET imaging in mice who were tumor-bearing. These are mice with syngeneic tumors, so they have a normal immune system. And then we separate the tumor while it's still hot. So this is the graduate students working around our lead castle. to separate across a magnet whole tumor from tumor-infiltrating lymphocytes, tumor cells, myeloid cells, spleen cells, and other cells. And then we just count radioactivity, very old school. And what we found was a little bit surprising to me because I really thought the cancer cells would win. And this is all in animals, but in a variety of models, all of those, the 4 across the top, in 2 different colorectal models, a kidney cancer model, a breast cancer model, and then you can see listed below orthotopics, GEMS, induced models, all had similar results, which is that compared to spleen in white, whole tumor uptake per cell in light gray, the cancer cells reflected sort of the whole tumor generally, that's in the dark gray, and red was immune cells. So on a per cell basis, the immune cells were preferentially taking up glucose. And so my husband thought, aha, he had won, it was the T cells, but it was not the T cells. And that's what you see over on the right, is when we further separated, the red is now macrophages. I think in this particular plot it's F480, but whether we use CD11B or any other myeloid macrophage marker, they are the winners in this glucose consumption. T cells are shown in blue, and the mixed immune cells are shown there in green. So I'm not going to show the whole details of this paper, but we were able to show that there's a pre-program for cells to select and use what it is that they want. That probably changes with time, and it probably changes with types of tumor. Here we're showing 2 different tumor models, though, where we said, well, what is the cancer cell consuming? And so we used F18 labeled glutamine. Same exact study. We did an F18 glutamine PET. There you can see the picture of the PET image and just an autorad of the glutamine to show that it's well dispersed across the tumor. And here, the results were quite different. Instead of showing up all in the myeloid cells, the F18 glutamine was showing up in the cancer cells, here for an MC38 model and in a RINCA model. And so if you think about the nutrients and how nutrients might be utilized, I'm just going to put it all up here. What we saw using F18 glucose, conventional FDG PET, was that it was immune cells, preferentially macrophages, who were happiest using this. But if we wanted to look at the cancer cells, at least in these models, it was F18 glutamine. In vitro, we used a fluorescent fatty acid probe to see if fatty acids were preferentially going one way or another. And at least it looks, at least preliminarily, that this is going toward the cancer cells, that the cancer cells were more favorably opposed to utilizing fatty acid. This is all in an untreated state. But I wanted to share the story because I think that it's important as we think about how do we use PET scans or other functional imaging to look at the whole tumor, if that's in fact what we need to do. And it begs the questions, are there features in an FDG PET that can help us know if we're looking at cancer cells or immune cells or a mixed signal, quantitatively or otherwise? Are there features of an image that are indicators of response or resistance? And are there better PET tracers? And we already had a beautiful talk on that. Are there other functional imaging tools? And can we accelerate strategies for bringing these more into patients? And so I'll just end with, I think for surrogate biomarkers, preaching to the choir, we need easily accessible, rapid analysis, quantitative, and that are systemically informative, whether that's imaging or blood-based work. So I'll stop there. That's my lab. Thanks very much.